So today I'll be talking about the replication fork in prokaryotes. I have a simplified DNA figure drawn with the 5' and 3' ends labeled, showing two strands running antiparallel in a double helix. I've drawn a zoomed in version of one strand, showing how we label the ends. The strand is made of sugars, bonded to each other through phosphodiester bonds, which are the PO4s. I've labeled the carbons in both sugars in red, and you can see that the top sugar has carbon 5 on top, and as you go down the strand, the sugar then connects to the next sugar via its carbon 3, which is why we say DNA runs 5' to 3'. I've also shown that the bases bond to each other through hydrogen bonds, which is part of what holds DNA strands together. In the next figure, I've drawn the origin of replication, which is a DNA sequence that is recognized by an initiator protein to denature the base bonds and form a replication bubble. Replication occurs in both directions from this origin. The enzyme helicase then is able to take over unwinding the DNA by breaking the hydrogen bonds between the bases. The topoisomerase gyrase then binds to the double-stranded parts of DNA and nicks the DNA to prevent supercoiling. RNA primase also binds near helicase and makes the primers that DNA polymerase will be able to recognize to start forming the new strand of DNA. Single-strand binding protein binds to the single strands to stabilize DNA in the single-stranded state and prevents hairpins from forming. DNA polymerase 3 binds to the RNA primers and starts to form DNA in the 5' to 3' direction. This means there will be a leading strand, which is able to continuously make DNA, and a lagging strand for each fork in the DNA. The lagging strand has to make the new strand of DNA in parts called Okazaki fragments, as DNA polymerase has to make DNA in the 5' to 3' direction, making it travel in the opposite direction as helicase unwinds the DNA. Multiple RNA primers are used to signal to DNA polymerase 3 to attach again closer to helicase and make DNA until it runs into the previously made fragment and jumps toward helicase again. The fragments from the lagging strand of DNA are separated by the RNA primers, which DNA polymerase 1 binds to and replaces with the correct DNA code. The polymerase enzymes have proofreading capabilities and read through the DNA for any errors as well. When the primers have been replaced with DNA, ligase binds the DNA fragments together to form one continuous strand, and the product of this process is two DNA structures with one old strand and one new strand.